today about my work on in vitro differentiation of human theca cells. As we know, theca cells are essential for follicle development and estrogen synthesis in the ovary. Despite their vital role in female fertility, the ontogenesis of these cells remains poorly understood. While it is believed that factors secreted by oocyte and granulosa cells are responsible for recruitment and differentiation of theca cells, we really do not know which cells are recruited to be differentiated into theca cells. Studies in mouse have pointed out to a population of theca precursor cells that are present in the ovaries. On the other hand, some interesting studies in cows and goats indicate that stromal cells from ovarian cortex can differentiate into theca cells. As human ovaries are similar to ones from large animal models, we hypothesized that under the influence of growth factors secreted by granulosa cells and oocyte, stromal cells from the ovarian cortex can differentiate into theca cells. To prove our hypothesis, we have isolated the stromal cells from the cortex of postmenopausal ovaries and divided them into two different groups, where we could separately assess the effect of the growth factor. So in this way, in the first group, we cultured the stromal cells seeded in plastic and using basic medium, and we use knockout serum as supplement. In the second group, we also had a stromal cells seeded in plastic, but we used an enriched medium, the basic medium supplemented with growth factors and hormones that are supposed to be involved in theca cells differentiation. Before starting the culture and after 8 days of in vitro culture, quantitative real-time PCR and immunohistochemistry on genes and proteins involved in theca cells' stridogenic pathway were performed respectively. We added two more markers, ADRP, the protein in the membrane of lipid droplets, and CD13, a specific marker for theca interna involved in steroid hormone production. We also analyzed the ultrastructure of the cells before and after in vitro culture using transmission electron microscopy. Finally, we measured progesterone and androstenedione levels in the medium at days 2, 4, 6, and 8 after the culture using ELISA kits. Going to our results, stromal cells seeded on plastic adhered to the bottom of the well after 1 day of in vitro culture in both G1 and G2. But after 2 days, spheroid structures were formed and some detached from the well, here called floating structures or FS. At day 4, almost all spheroids detached from the bottom of the well in G2 and continued growing, while fibroblast-like cells, here we call them AC, remained strongly adhered to the plastic well in G1. Such spheroid-like structures were large in G2, and sometimes they measured almost 300 micrometer. qPCR analyses are shown in volcano plots, and x-axis represent fold changes in gene expression after 8 days of being in culture versus day 0 before putting the cells in culture, and y-axis represent logarithmic p-value set to 0.05. Genes above the dashed horizontal gray line are significant with p-value less than 0.05. and under this line are not statistically significant. Although these two cell groups in G1 show different mRNA and protein expression involved in the steroidogenesis of theca cells, they were both significantly positive for CYP17A1 mRNA, which is marker for theca cells. This made us to believe that cells were indeed differentiated into theca cells. As you see, for the immuno results in attached and floating structures, almost all the genes involved in the steroidogenic pathway, these proteins are expressed. We were very astonished by these results, and we decided therefore to perform CD13. And as you see here, CD13 for attached structure is 2%, and for floating structure is also 2%, the marker for theca cells, so it is not expressed. So How these cells look like through TEM? Well, we observe round and elongated mitochondria and gap junctions between cells in AC1, while cells in FS1 had only round mitochondria. In both AC1 and FS1, some endoplasmic reticulum and lipid droplets were detached in examined cells, which is an indication of hormone synthesis. Indeed, media collected in G1 show synthesis of hormone, a significant increase in secretion levels of DHEA, but not progesterone after 8 days of in vitro culture. If cells have enzymes involved in a steroidogenic pathway, but they are not theca cells, what are they? Well, increasing secretion pattern of DHEA and no progesterone during in vitro culture, and specifically presence of gap junctions in attached cells in G1 indicate that cells in this group have differentiated into ovarian interstitial steroid-producing cells. What about cells in G2 where we added the growth factors? So, in G2, where we added growth factors and hormones to the medium, significant upregulation for HSD3B2 in attached cells and floating structures is observed, plus significant upregulation for LH receptor mRNA in F2. Since most of the cells in G2 were forming floating structures, we couldn't get enough attached cells to perform immunohistochemistry for all the markers. So we decided to choose the most important mentioned as a specific marker for theca cells in the literature to carry on the immuno. So CD13 and CYP17. Our results show that while a significant proportion of attached cells differentiated into theca cells, the proportion of CD13 positive cells from the floating structure was not different from the control. Ultrastructure analysis from G2 evidenced that attached cells had a round, oval, or somewhat undulated nucleus, rough and abundant smooth endoplasmic reticulum, Golgi apparatus, lipid droplets, oval mitochondria with tubular or vesicular crista, and lysosomes, which matches to the ultrastructure of human theca cells according to the literature. Ultrastructure analysis from F2 cells revealed the floating structures contain 3 different cell types. Fibroblast-like cells with elongated nuclei marked with circle here, and transitional cells which are ellipsoidal in shape with an oval nucleus, and here they are marked with a 4-tailed star. And theca-like cells which have the same ultrastructure as described for attached cells in G2 with undulated nuclei, and they are marked with 5-tailed star. In G2, medium analysis revealed an increase in DHEA and progesterone synthesis during in vitro culture. I've included these graphs with both G1 and G2 so we can have a better idea regarding the synthesis of hormones. By looking into the graphs, we see a tendency for higher DHEA secretion in G2. We also observe a statistically significant higher secretion of progesterone in G2 compared to G1 at day 8, which indicates that the active theca cells. For more information, there is no granulosa cells in G2, so progesterone is not transforming into estradiol. To conclude, it is possible to differentiate human ovarian stromal cells into theca cells in vitro. Growth factors secreted by oocyte and granulosa cells are involved in theca cells recruitment and differentiation. And if all the stromal cells can differentiate into theca cells, most of the cells differentiated into interstitial-like cells, while a proportion of them, 43%, differentiated into theca-like cells. This can lead us to the presence of a subpopulation of theca cells in human ovaries, and the studies in cow and goat could be a mix of theca cells and interstitial cells since they haven't studied CD13. Thank you.